All right, so our next speaker is Zhengxu Lian. And Zhengxu is a third-year PhD student at the Institute of Science and Technology for Brain-Inspired Intelligence, Fudan University in China. And his research focused on body-brain interactions and their roles in psychiatric disorders. He is supervised by Professor Jian Zhang and is committed to uncovering multimodal biomarkers that bridge physiology and mental health.
So thanks for your introduction, and hello everyone. Thank you for being here. I'm Zhengxu Lian, a third-year PhD student from Fudan University, East Bay, China. Today I will talk about our recent work, where we look at how depression is linked to physical health, especially metabolic disorders, by combining brain imaging and plasma protein data. So we all know that depression is common. It affects about 50% of the population. But what makes it so complex is that it's not just a brain disorder, it's also deeply tied to the body. One piece of the puzzle is plasma proteins, which reflects what's going on systematically in the body. Another key piece is brain structure and function, which play a major role in how depression manifests. So we ask, can we find a shared pattern between plasma proteins, brain imaging features, and depression? Something that might help explain how the body and brain are connected in this disorder. To tackle that question, we use the multimodal fusion method called MCCR plus GSA. In simple terms, multimodal canonical correlation analysis with reference finds features in each data modalities like brain volumes or protein levels that are linked to depression. Then joint independent component analysis further separates the remaining mixture in the joint maps. We apply this to 6 brain imaging modalities and 1 proteomic dataset. This approach helps us find joint patterns that cut across body and brain. We work with data from the UK Biobank using almost 4,000 patients who had both brain imaging and plasma protein data. So the study has 3 objectives. First, we identify the main multimodal component, what we call neuroprotein. Then we tested this stability in other datasets, HCP and Southwest University depression dataset, where only brain data were available, so we only test the brain modalities. And finally, we dug into the biology, looking at the environment, lifestyle, and genetic factors that might explain the patterns. So what did we find? The neuroprotein component clearly separated people with depression from healthy controls across all brain measures and protein profiles. And the key brain regions included the sensory motor cortex, temporal lobe, thalamus, hippocampus, and especially the subclavicular gyrus, which is already known as a target for depression treatment. And it also has some metabolic alteration in the depression patients found previously. And we also show a unique pattern that asymmetry in the superior temporal circuits and also find some proteins related to metabolism, immune function, and neuroprotection. Some like GH1 and IGF-BP1 are linked to obesity and insulin resistance. We also look at how these brain changes relate to neurotransmitter system. For example, functional connectivity changes were tied to serotonin and dopamine markers like SERT and others. We saw structural connectivity linked to serotonin receptor like 5-HT1A and 5-HT1B. So to check how stable this biomarker is, we tested it in 2 external data sets and found that it could still predict depression symptoms quite well. This shows that neuroprotein generalize across data sets and is biologically meaningful. Next, we explored what these proteins actually do. We use proteins modality from that joint component and using FUMA. We found that they are mostly expressed in the pancreas, liver, blood, and brain, which really supports the idea of body-brain interaction. And functionally, they were involved in metabolism, immune response, and insulin-like growth factor signaling. So again, we see metabolism showing up as the major theme here. We also use network analysis to see how these proteins group together into co-expression modules. Interestingly, proteins that show up in the neuroprotein clustered into metabolic-related modules where those folk found using the standard regression were more immune-related. This suggested that our method may be revealing a slightly different biological signature, one that's more metabolic than what the univariate regression analysis approaches typically capture. We also ask, what else are these proteins of neuroprotein related to the real life? So we found links to the air pollution and environmental exposures, diabetes and insulin use, alcohol consumption, and body impedance, which reflects the metabolic health. This tells us that neuroprotein may capture a subtype of depression that deeply tied to both lifestyle and metabolic condition. So we took this one step further and look at the physical diseases. So we used the previously defined neuroprotein proteins, and they were associated with some polygenetic risks, like for type 2 diabetes, body mass index, and chronic kidney diseases. And we also want to ask if they can predict the future onset of some physical disease. And we found like hypertensive kidney disease, atrial fibrillation, and pancreatic disorders. So beyond just explaining the depression, neuroprotein may help predict combined conditions as well. Building on the disease association we just saw, we want to understand the mechanism behind these links, especially the role of metabolism. So we use body mass index as a proxy for metabolic status, and since it's strongly tied to insulin resistance, inflammation, and overall energy balance. So we use monoline randomization show that these proteins don't directly lead to the depression, but lower protein levels lead to the higher BMI. I was put there the lower protein levels are observed in the depression patients, and this which in turn raises the depression risk. So to warm up, neuroprotein deep is a robust biomarker that ties together brain features, plasma proteins, and depression. And it's point to metabolism as a key player in the body-brain link. And this opens up the door for metabolically targeted interventions and might help prevent secondary diseases in people with depression. And finally, it shows the power of combining brain imaging with proteomics to uncover new types of psychiatry biomarkers. And thank you so much for listening. I also want to thank for my extraordinary colleagues and supervisors. Thank you.
So we have an online question regarding your methods using MCCAR. So this person wants to know how stable do you think this method or this solution is? Have you done any validation tests like test retest or split half?
Yes. Firstly, we done in dataset validation split half test, and also we use two external validation sets, but due to the unavailability of both plasma protein and brain, we only did brain test validation. Okay.
Okay. I have a personal question, more or less. So in your almost last part, you did the mediation analysis, trying to see whether any effects go through the BMI-related proteins to the downstream behaviors. So I'm curious because the BMI or inflammation or metabolism have come up in a lot of other psychiatric disorders. I wonder about your thoughts on how specific your results here are to depression or any other related disorders.
Yes, this is an interesting question. So what we hear is only show that maybe the metabolic is a path to the depression, but I also think that there might be other psychiatry disorder show the same pathway, and we are now have our future work in the term of this direction. We use the multi-omic biomarker to show the heterogeneity of psychiatry, including the depression.
Okay, thank you very much.
Thank you very much.